Hereby, I open this academic ceremony in which Patrick van Zalm will defend the academic thesis, Development of Large-Scale Methods for Body Fluid Proteomics and their Application to Biomarker Discovery in Alzheimer's Disease. May I invite you to present a summary of your study and the conclusions of your thesis?
So, thank you committee for joining me today. Thank you promoters for joining me today. Thank you Hanno and family and friends here all. So today I would like to present you my thesis called Development of Large-Scale Methods for Body Fluid Proteomics and their Application to Biomarker Discovery in Alzheimer's Disease. So if we think about the disease Alzheimer's disease, this is a disease that mostly affects elderly, and they suffer from memory loss, difficulty of thinking, poor judgment, as well as disorientation and others. And of course, it's a high burden on the patient itself, but of course also society. Think about caregivers that have to help those patients, or just the medical cost involved, right? Currently, we know there's roughly 50 million patients across the globe, and this number is expected to increase to roughly 125 in the next 30 years, right? Making it a very big problem. Now, if we think about the disease in a more biological way, what we know is that the healthy brain on the left kind of over time shrinks to a smaller version, which is of course caused by brain cells or neurons dying over time. And this dying of brain cells is caused by many different biological things, right? The most common ones that are known are the amyloid plaques and the neurofibrillary tangles. But there is of course more out there. And it's a bit poorly understood how they interact with one another, which ones come first, but they are known to all play some kind of a role in how the disease progresses. If we think about the detection or finding out if a patient actually suffers from disease, is that on the x-axis we can see how the clinical symptoms move over time. And these clinical symptoms here are drawn in purple and green, right? The issue is that if a patient is, let's say here in the middle, it means, or it's known that years in advance, this pathology or this kind of damage already started occurring. So if you would detect a disease here, you have the problem that you already have years of damage in the brain. This is quite problematic because this makes treating the disease difficult. And so what researchers would like is that they are able to not detect the disease when the clinical symptoms occur, but instead when, as early as possible, right? Preferably when the first pathological or biological hallmarks start happening, or maybe even earlier, right? That is the big goal we want to go to. And there's multiple groups working on this. There's multiple, researchers across the globe trying to do this. There's different ways to do this, but we, doing protein research, believe proteins can be the solution to find out about this. So then, of course, the question arises, what are proteins, right? Now, for a lot of people, it might just be as simple as an egg, right? But when we think about proteins, we think about something else. So you get your genes or your DNA, one part from your mom, one part from your dad, and this DNA, right, defines your genetics. Genes are then translated in your body into proteins, and in science, we like to draw them like this, quite difficult to read. But what the point here is that proteins can play all kind of roles in the human body, right? It can be literally anything, such as light coming into your eye, being transformed into an electrical signal going to your brain, or the enzymes that digest your food in your stomach, also proteins, or just the movement of your muscles that makes you able to walk or do things. All in all, they're all proteins. And what we do in proteomics is try to identify and quantify as many of these proteins as we can. And with that information, we try to answer biological questions. A proteomic study has a quite common workflow. So what we do is we have a biological sample. We extract proteins from this sample. So we want to get rid of other stuff that's inside of that. We cut them into small pieces. And then we use liquid chromatography to separate them over time. These small protein parts are then injected into the mass spectrometer, where we charge the mass to charge ratio, or just the weight. Then we need to figure out what this smaller protein did belong to originally, and for that we have computational algorithms that help us to do that. And then finally, with that information, we can try to find information about specific proteins related to a disease or maybe biological pathways. Now, this is, of course, a proteomic study in general, but if we want to do it in a more clinical application, we believe that especially body fluid proteomics is of high interest. And there is different aspects related to body fluid proteomics, such as the extraction, extracting the information from the data that we generate, or other statistical analysis. And this field has made quite some big steps in the last couple of years. So 10 years ago, we could do maybe 5 or 10 or 50 samples. But in the last 5 or 10 years, we have now done hundreds or thousands, and I guess 10,000s or 100,000s are numbers we will see soon. And if we start doing these large-scale studies, it means we can overcome some of the statistical barriers that come if you work on smaller data sets. So this progress, right, of being able to do large-scale proteomics has been the work of many researchers, and I believe that some of the chapters here in this thesis has also been important for the field itself to make large-scale proteomics possible. For example, in the second chapter, what we kind of tried to create was an improvement or improved method to extract proteins from blood. Because when we think about blood, one of the issues if you work with blood samples is that just a few proteins account for up to 99% of all the protein content in a given sample. And this makes identifying proteins that are of low abundant range quite difficult. And of course, these are the proteins you're interested in, because of course you're thinking about Alzheimer's, right? You would expect the proteins would come from the brain. and therefore they will most likely be in this low abundant range. So we want to reduce down the effect of these large-scale proteins. So to do that, we developed an acid precipitation method, and it roughly looks like this. What we do, we have a blood sample being collected from a patient. It is spun down to create something called plasma. The plasma is added to a microtiter plate, it's called, so you can do a lot of samples in a small box. Then on top of that, we add the acid. This creates a kind of separation of the body fluid. It's spun down, and then the supernatant, the part on top, is collected and injected in our analytical instrument. And this is a study we have done with thousands of samples by now. And what I think is interesting to show today is that when we do not use this novel precipitation method, is that we can identify 59 proteins that are currently approved as a clinical detection method by the FDA in the United States. But if we use our developed acid precipitation method here in red, we get an additional 18 of these proteins on top of that. So it clearly shows that it's an excellent depletion method. We've done it on thousands of samples, so we consider it high throughput and robust, and also importantly, it's quite cheap. In the next chapter, we can create data now of thousands of samples, but of course, handling such data also requires some new methods. And this is work that has been published in a Journal of Proteome Research. Because if we collect data on the mass spectrometer, on this analytical instrument, what we kind of get to is this figure that you see here, the mass to charge ratios. What you need to know is that computers figure out which protein this originally belonged to. And of course, if we have 5 or 10 samples in the computer, it's fine. You can get it done. But if you have, let's say, 5,000 of them, you would think, okay, let's buy more computers. But eventually, this does not become feasible, right? You can't run around, start computers, and so forth. So instead, what we wanted to do is to use high-performance computing. This is also commonly called a supercomputer. So we kind of redeveloped the method there, where we especially try to apply parallelization methods to facilitate the speed of that. And for example, one of the workflows that was involved in here, we were able, with this high-performance computing and this parallelization, to reduce the time from 93 days down to less than 10 hours. All in all, we were able to do it in less than 10 days, and it really showcases that high-performance computing is an excellent way to do these large-scale proteomic studies. In the next study, we wanted to create some statistical methods to really do population-sized studies for proteomics. And this was recently published in Cell Report Medicine. One body fluid that is very interesting for researchers that do Alzheimer's research is the body fluid cerebrospinal fluid. It surrounds the brain. It's the closest to the brain. So if there is proteins that give information about the disease, this is the body fluid you would expect them first. So of course, researchers across the globe have done this, and what we noticed there is that the results they show can be actually quite variable, which is weird, right? Because they all study the same disease. So we figured, okay, it could be due to variability in the machines used, statistics used, software used, and so forth. So maybe a solution would be to really redo it all systematically. This is what we did. We re-downloaded the data, we reanalyzed it completely using the same methods. And what we found with roughly 150 samples is that we saw an upregulation of proteins related to energy and metabolism, right? So really the energy pathways in the human body. Now we figured, okay, this looks good, but we want to validate it, right? So we had an additional in-house data set, and here we saw that of these initial 21 proteins, 3 of them proved to be, again, upregulated in Alzheimer's patients. Okay, we validated it once. Let's try it again, right? We re-downloaded 2 additional large-scale data sets, each comprising at least 100 samples. And there again, we saw a high upregulation of these proteins. So all in all, in this study, we were able to do a first-of-its-kind meta-analysis of raw proteomics data. We noticed metabolic changes in Alzheimer's, and this really showed to be a validation of biomarker candidates for Alzheimer's. And then in the final chapter, right, what I just showed you was cerebrospinal fluid. And And this is, of course, a great body fluid if you want to get an idea of what happens with Alzheimer. But if you want to collect it, it's quite difficult. So you need a medical specialist that puts a needle in your spine. It's not very easy to do. You couldn't do this on thousands or 10,000s of patients on a daily basis, right? It's just not really feasible. So instead, we believe that using blood or plasma would be a better candidate for such application. The reason for this, of course, every hospital already does it. It's very common. I think most of us have given blood at least once in our life, so it's easy. And so we had a data set comprising of healthy people. We had patients suffering from early Alzheimer's disease, so really at the earliest point, and then something called mild cognitive impairment. This is this stage before Alzheimer's when they might develop the disease, but also might not. It's a bit before. So we first analyzed this cerebrospinal fluid, and what we noticed here is that each dot here represents one patient or one person, is that especially here we noticed that the proteins in the cerebrospinal fluid behaved quite similar for the mild cognitive impairment and the Alzheimer's compared to healthy patients. So we could find damage, but we could not maybe make the distinction between these two diseases. Then, when we used blood, what we noticed is that there was actually a very clear distinction of these Alzheimer patients, but not these mild cognitive impairment patients, right? So all in all, I think this is a great case where in a relatively small data set, plasma can detect a disease like Alzheimer's disease, even when these proteins might come from the brain. And all in all, I think that in this thesis, we have really done work that make large-scale proteomics possible for the future. And if you think about Alzheimer, if we really get to a point where we can start analyzing thousands or 10,000s or even 100,000s of patients suffering Alzheimer's or other types of dementia, maybe then we can overcome the barriers, the statistical barriers that come with smaller data sets. And maybe only then we are able to find these detection characteristics that make it possible to detect the disease not when a patient shows symptoms, but instead even before they could even know. And if we would that, I hope and believe that we can prevent the brain from turning from this healthy left brain to this diseased right brain. Thank you.
Thank you very much for your presentation. The opposition will now be opened by Professor Raamakers, Professor of Psychopharmacology at Maastricht University, and he was chairman of the dissertation committee.
Thank you. Dear candidate, first of all, I would like to congratulate you with your thesis. It was really very interesting to read. It was well written, it was condensed. And in a way, it also reads like a small success story, right? In the sense that you have developed this method for a biomarker of Alzheimer's disease. And you were also able to validate the method as such, which is quite an achievement, I think, which you have done in almost 4 years. So congratulations with that. And also, of course, I extend these congratulations to your supervisors. And it's also nice to see a student from our drug development and neurohealth track, right? So we have seen you coming in, we've seen you develop into this stage where you actually are right now. So that's quite a good achievement. But coming back to your method, right? One question I had was whether this was a lucky shot, in a sense. So why did you choose proteomics, right? In your presentation here, you already made clear that there's more than just proteins. There's metabolic activity, there's energy levels. So why then did you focus on proteomics rather than genomics or metabolomics that maybe would have given you the same outcome as, maybe it would have even been more selective? I couldn't really find in your thesis a rationale up front why you chose for proteomics rather than any other method that maybe would have been as effective or successful.
Okay, thank you, highly esteemed opponent. Right, I think if we think about diseases, they are of course caused by the pathology, and in the end, I believe that proteins cause this pathology, right? There is genetic defects that might not always cause disease and could also be the other way around. So, yeah, if you think about a disease like Alzheimer's disease, right, we know that the most famous ones, amyloid and tau, right, they are also caused by proteins. So if you ask me, coming from proteomics, I believe that measuring proteins would be the way to do this, right? I mean, if you look at genomics, you could have the E4 one from amyloid and still not develop the disease, right? It's a risk assessment at that point. So,
but they would also be involved in metabolomic pathways, right? So wouldn't that indicate that, okay, you can use proteomics, but would it be more sensitive than, for example, metabolomics? Or would you predict that if you use that metabolomics method, you could develop a similar biomarker?
If you ask me, right, I think that if you really want to detect a disease as complicated as Alzheimer's, right, a disease that develops over years, if not decades, it will most likely really be a combination of biomarkers, right? And well, I really think that proteins will play a major part in this, but I'm also not stating that it will only be proteins, right? Indeed, maybe there is a combination of, right? I can imagine that the genomic information about amyloid will already help do that assessment, and then combinations of proteins and or metabolites will make it really possible to do such detections of such a complicated disease. So I'm not saying that a method such as metabolomics is not as good. I think we should more consider them complementary to one another, right? So proteomics, metabolomics, transcriptomics, lipidomics, all of them could be the hidden key to detect such a disease. And, well, we are doing proteomics, and this is the method that we are experienced in. But I think really in the future, it could be a combination of them.
Okay, so indeed, that's future music. I was also thinking about your invention from the perspective of the patient. I was seeing myself going to, I don't know, general physician who's using your method, and then I would get the message, well, Professor Ramakers, we've detected early Alzheimer disease. It's not MCI for sure, it's early Alzheimer disease, but that would be it, right? But then I would have information about a disease that we cannot treat. Mm-hmm. So from a patient perspective, I'm not so sure what the advancement actually is of this particular biomarker. I see the technical advancement, that's quite clear. But what I wonder is whether your method, your biomarker, actually also tells us something more fundamental about the etiology of Alzheimer's disease, and whether this could actually help us rethink the causal mechanisms that underlie Alzheimer's disease.
Yeah, I mean, I think for developing medication for any kind of disease, a biomarker is of great worth, right? Being able to determine if a potential pharmaceutical or other type of intervention really reduces the burden of the disease is worthwhile information. And I think more specific to Alzheimer's, right? As we know, right, that once clinical symptoms start occurring, a patient already suffered years of neurological damage at that time point, right? And I think with such a complicated disease, it is quite worthwhile to know or to detect this disease as early as possible. And indeed, initially, it will maybe not be worthwhile to a patient itself. It could maybe even considered negative, right? I guess it will also create some psychological burden. But I think we should more think about the future, right? Because if you can really start detecting this when the amount of damage is the bare minimum or even, well, let's say 0, maybe then you can already start creating medication that prevents the pathological development over time, right? So I think really to reduce that, it's important.
Yeah, I would agree. I think that would be very important. But then if you look at the proteins that you've identified as biomarkers, would you see them as, let's say, symptoms of the disease, or do you really think, okay, these are also proteins that we need to target with medications, for example, somehow to prevent the development of, or to stop the development of any of these cognitive declines. How do you see that?
I mean, I personally, if you ask me, right, I think that, again, it's a very complicated disease with different pathological hallmarks, all having a role in the development of the disease. And of course, as some companies across the world, right, they try to develop antibodies for amyloid or tau, right? And these have not really proven to be, the key to solve the disease, right? So if you ask me, I guess there is more things that need to be solved to really stop the disease from progressing, or at least reduce the burden. And then something like metabolic changes or the dysfunctioning of calcium homeostasis, right? I think all these little chapters that define the disease or develop the disease should be solved. And yeah, this will most likely be, I guess, a cocktail of medication at different time points. But I also don't see another solution at this time point. So,
well, I'm not saying I'm looking forward to this cocktail, but thank you for the answer.
Thank you very much. The opposition will now be continued by Professor Oerlaub, Professor of Bioanalytics at the Max Planck Institute for Multidisciplinary Sciences and University Medical Center of Göttingen. And he is also a member of the dissertation reading committee.
This one? Okay. Yeah, dear candidate, thank you very much for this clear and very good presentation, and also congratulations from my side for these excellent results. As far as understood, this is the largest data set that was obtained so far from a study. Also congratulations to the supervisors and co-supervisors. Excellent study. Of course, I have also several questions. Is there any definition for proteomics? Can you put this in one sentence? What is actually
yeah, thank you for the question, highly esteemed opponent. In my head, the definition would be the identification and quantification of proteins, peptides, their structure, and their post-translational modifications in a biological sample.
in a given state?
Sorry?
In a given state.
Depending on the question that a researcher wants to answer. Yeah. Yes, of course.
Good. And what is the definition for the term protein? Do you know that?
Polypeptide, if you want to go.
Where does the word come from? You know that?
Multiple peptides connected, right? So multiple amino acids that are connected to one another.
It's actually from the Greek word proteus, of the first rank. It was discovered first. I'm also the opinion that the proteins are of the first rank because they reflect everything what is happened in the living organism. On page 11 of your thesis, you raised this question whether the aggregated proteins are the cause, or, as Professor Rehmaker said, the symptom. What speaks for the cause? What speaks for the symptoms? Could you summarize this?
Sorry, could you repeat your question, please?
So you have the amyloid beta plaques and also the tau fibrils in Alzheimer's disease. Is this the symptom, or do they cause the disease? Could you argue in both ways, maybe?
I guess that's the million-dollar question to some
degree. Yeah, of course.
I personally think that, Frank, I don't know, to be fair. I personally, if you ask me what I think about it, is that I can imagine, right, that damage over time, such as oxidative stress, that they could maybe influence biological pathways over time, and that ineffective other bodily functions or other biological functions might, for example, cause this hyperphosphorylation of tau, right? Because this hyperphosphorylation of tau is the cause of these neurofibrillary tangles forming, or the wrong cleavage of the amyloid protein, creating these amyloid beta plaques, right? So if you ask me what I think there is something up front of these two main characteristics. But what really is the cause? That is, of course, difficult to answer, right? The biggest cause is aging. But yeah, how do we prevent that, of course?
Yeah. I mean, these proteins somehow accumulate, you said. How is the protein homeostasis in general regulated in a cell? That you keep the levels of protein constant, more or less.
Across all time, right, proteins are oxidated usually, which means they are kind of signed as, they should be removed from the cell. Then cleavage pathways could reduce them down to smaller parts, which will then be removed from the cell and eventually, leave your body.
Yeah. So there's the proteasome plays a major role in the cell, right? Because it degrades proteins. Proteins have to be marked to be degraded. Might there be some changes in the proteasome, in the activity? Has this been investigated? Do you see?
Specifically for Alzheimer's, or?
Pardon?
Specifically for Alzheimer's, or?
Yeah.
Yeah, there has been papers out there that try to research, right, the proteosomes related to Alzheimer's. I'm not completely up to date on the recent literature there, but it has been done, and I think it's also one of the fields. Yeah, there's still a lot of work to do, especially for a disease like Alzheimer's.
Yeah, absolutely. In this respect, you observed that the enzymes for glycolysis were upregulated, right?
Yes.
What does this mean, actually?
So indeed, right, it's a quite interesting result that you find upregulated proteins when you would maybe expect downregulated proteins, right? My first idea was, okay, I would expect the proteins to go down if we consider pathology. Right, but this indeed could be they could be indeed the result of this affected proteases that might clear out more of these proteins from neurons, which would of course then end up in the cerebrospinal fluid, and therefore they inversely would lead to a higher abundance of such glycolytic proteins.
Normally, when you have more glycolysis as compared to oxidative phosphorylation, then this means that mitochondria do not function very well, right? Do you have any clues about the mitochondria proteome in your studies? Or whether the TCA cycle is affected?
I think this is difficult to answer with the proteome of cerebrospinal fluid, right? To really make that distinction clear where, for example, the proteases happen.
Yeah.
I think that distinction we can't make with the current techniques we have.
Okay. Do I have time for a third question? One question, thank you. So a very important step in your sample preparation was the protein precipitation, right?
Yes.
This is indeed of utmost importance for any proteomic workflow because you need to concentrate your samples or you have to remove something. Why does this work, your precipitation step with the perchloric acid, especially for that you get rid of albumin? Is there any physical-chemical explanation for this phenomenon?
We believe that the high pH level that you add to the plasma or serum or body fluid sample that this incredibly high pH level really affects mostly these high abundant proteins. To be fair, I think we have still not completely figured out why mostly these high abundant proteins are affected.
Yeah.
But we do know it's effective. We have tested it in multiple cohorts, in multiple studies, and we have also tried studies without this precipitation method, and we see the same biological pathways to be affected, right? So, we believe the method is solid, but the
no
actually
doubt.
chemical key behind why it works, we don't know yet.
Okay. Thank you.
Thank you very much. The opposition will now be continued by Professor Heren, Professor of Molecular Imaging at Maastricht University, and he was also a member of the dissertation reading committee.
Thank you, Mr. Chairman. Dear candidates, I would like to join my colleagues in congratulating you with your beautiful thesis, some wonderful work, and I'm also very happy to see that as a Maastricht graduate, you ended up in an international situation, and that just shows you how international science is and should stay in spite of what we hear in the media every now and then. As I said, I enjoyed reading your booklet, but we read it in the digital form when we did the assessment, and then I got this booklet, and I was shocked to see this list, particularly in proposition number 8. And I would like to ask one of your paronyms to read out proposition number 8.
Of course. New instruments, acquisition methods, and computational tools mean that in the next few decades, proteomics will join genomics and transcriptomics in becoming a reliable source to improve our understanding of biological processes and pathologies.
Thank you very much. I guess you understand where my shock comes from, because apparently you imply that proteomics, the work you did in your thesis, is not yet reliable. So my question to you is, what are your thoughts on that? How reliable are your results actually, if it will take another few decades before the method you used will become reliable?
Yeah, thank you, highly esteemed opponent. Indeed, good question. I think I'm speaking here from the point of view of large-scale body fluid proteomics, right? To really answer or to find detection methods for multiple diseases or other clinical states a researcher might be interested in. And yeah, as I kind of tried to point out a bit earlier or in the thesis, is that we come from a time where we could do maybe 5 or 10 or 20 samples in a given study, right? It would take a week of acquisition before we even have the data, heavily fractionated samples and so forth, right? And I personally think that, these have been quite helpful to detect the first signs of a disease. But as I think also research shown, most of these proteomic studies have not really led to any additional biomarker candidates that are in clinic used.
But as you show in your first introductory chapter, actually there's a lot of similarities between these, let's say, less reliable study. They still come up with roughly the same biomarker panels that you also find. Yeah. So what is then needed in your mind if you say, okay, large scale, the scalability of the method is maybe less reliable, and you've made a great step towards that. What in your mind is needed, and the experience that you have now, to become that reliable, high throughput technology that you look for to come up with these validated biomarker panels? What is missing?
So I think, so let's say in the future, right, or from now on, right, let's say hypothetically we would analyze 10,000 Alzheimer's samples or other types of dementia, right? There are so many confounders in a given data set coming from the clinical data, right? Of course, we know maybe their clinical definition done by psychological assessments, but these do not always completely correlate with what we see in, for example, a PET scan or measurement of the amyloid proteins through CSF. And of course, all these little, all these data points will require a lot of data to clarify how these confounders interact with one another. And this
But I think we're digressing a little bit from the point that I'm trying to make, because what you talk about now are clearly all the confounders that influence the outcome of a study. But in your proposition, you talk about a method, not about the confounding factors. So what is needed methodologically to actually make these methods, the new instruments, acquisition methods, computational tools, better and reliable?
Yeah.
I mean, understanding the confounding factors, of course. And I'll come back to that in another question. But I'm still sort of guessing. I thought proteomics was pretty reliable already.
Sure, but I think there is always room for improvement, right? Like anything. I think there is great methods or analytical instruments out there that have made such more robust methods possible, right? Think about an EvoSep liquid grammetry system, right? That is considered way more robust than some of the previous systems we used. You know.
Do you think, just out of curiosity, do you think the Astral will help?
Remains to be seen, would be my answer.
That's a good answer, I think. All right, let's drop this topic and move on. You actually mentioned confounding factors. And on table 5.1, on page 94, you talk about the 3 different cohorts that you've studied in chapter 5, where you looked at Alzheimer's disease, the mild cognitive impairment, and your control studies. And you find that there's quite a bit of difference between the MCI group and the Alzheimer's disease group in the end when you look at the plasma samples.
Yes.
But when I looked at the makeup of your cohort, I found something unusual, and that is that the Alzheimer's disease are predominantly male subjects, whereas the MCI group is roughly 50-50 gender balanced. Can you argue that this is what you're seeing, the difference that you're seeing is not a gender effect as a confounding factor, and that it's effectively not the disease state, but simply a gender effect?
Yeah, thank you for finding that detail. Indeed, right, there was a clear disbalance of the genders in the Alzheimer's patients. As we work with a relatively small data set, the most simplest solution to confirm our results was to just remove one of the sexes from the data set. And of course, since we reduced the amount of samples, our statistical power was reduced, right? So we found less proteins to be significant, but the majority of them did overlap with the initial results we saw. So for a first try of doing plasma proteomics for a disease like Alzheimer's, we were still very happy with the result we saw. But indeed, it would again require a lot more data to really make a strong statement in that sense.
Did you, for instance, also have a look at just the MCI cohort and then see if there's a separation between, for instance, male and female patients that you've included? Because that might provide a hint that there is or is not a gender effect.
I think I should do that then. Yeah.
I think that would be a good idea. Mr. Chairman, do I have time for one more question? One more, okay. Then I'm going to move you into methodology. You discuss in several parts of your thesis data-dependent analysis and data-independent analysis. And let me first ask you, sorry, I had only one question. Oh, no, this is getting very difficult. What method do you prefer for the purpose of biomarker discovery, and why? And if it's one or the other, why in chapter 5, I think it was, where you compared CSF and plasma. For CSF, you use both methods, and I think for plasma, you only use DIA. So, and is that still a fair comparison? Is that not comparing apples and oranges?
So, to answer the first question, which has my preference, it would be DIA. I prefer the data-independent acquisition method more. The reason for that is that, of course, we don't have the machine doesn't select the top 12, so it might remove potential peptides we're interested in. Of course, this brings quite complex data, but I think with the tools that are becoming more and more, standardized and robust, I think that this is not really an issue anymore, if you ask me. And then for why we did both in the CSF and only DIA in the plasma. We have done a lot of plasma proteomics in Hanno's lab, right? And at that time point, we had acquired thousands of DDA plasma samples using the same LC method and the same MS method and the same precipitation method. And for us, there was really no need anymore to acquire more DDA data to increase our library, right? I did indeed acquire this data, actually. It's not written, but I tried it. It didn't really add anything. Instead, with the CSF, of course, we do not have such an extensive library, and therefore we did also acquire DDA data, which is then described in the thesis.
Clear. Thank you very much. I'm very happy with your answers. And I'll give the word back to the chairman.
Thank you very much. The opposition will be continued by Professor van den Hove, Professor of Neuroepigenetics at Maastricht University, and he was also a member of the Dissertation Reading Committee.
Thank you very much. Let me first of all start to congratulate you with a very nice thesis, dear candidate, which was really a pleasure to read. I'm always happy to see FN or DN students make it this way. Some questions. Let me also first start with chapter number 5. I will not touch upon the sex effects, but I was wondering whether in the end you were happy with the, let's say, apparent discrepancy between the CSF and the plasma data. So in the CSF, you could see that the MCI and AD cases were similar and could nicely be discriminated from the controls. When you then go to the plasma, which you also see as a less invasive tool, then actually you could particularly discriminate the AD cases from the other groups. So of course you explained that in your favor in the discussion of that chapter, but what are your real thoughts about that, on this discrepancy? Was this the ideal result for you or not?
Yeah, thank you for the question. A highly esteemed opponent. I guess in an ideal world you would have a perfect PCA plot with one cloud of AD samples, one cloud of MCI samples, and one cloud of healthy controls or whatever additional disease or group you add in it. But in my experience, you barely get to these kind of results. What I do think is interesting to note is that as it has been described, that these pathological changes happen before we even notice clinical symptoms. I think it is quite promising or interesting to see that the proteome between MCI and AD was not that different from one another. So this is, I guess, also kind of what you would expect. Also, some of the proteins that were one of the major factors to create this PCA plot were proteins such as tau or neurofilament light or some of these well-known ones. For plasma proteomics, especially for Alzheimer's disease, there's not that many studies out there that have done it. So, of course, you're a bit blinded in that sense. You don't really know which proteins to expect there. Yeah, I agree with the point, right? It would be the most beautiful if they would all three show a clear separation. And I mean, I'm not saying that the MCI is not completely overlapping with the healthy controls. There is a little bit of distinction there, especially in the PC2. But of course, the strength of that component is not that high. And then, of course, you get back to the point, I guess we need larger studies to really make those distinctions or to identify and characterize these distinctive features.
I agree, because indeed it would be very interesting with this less invasive method when looking at the plasma, that you can actually already pick up the MCI cases and discriminate them from the absolute controls. But that's indeed probably a lack of power in this case. Alternatively, of course, part of this could be explained by the sex effects, as was touched upon before. So maybe move to chapter number 4. And this was also already discussed briefly, and where for solid and valid reasons, I think you focus particularly on the technological aspects of your approach, as well as on the computational part of it. And you focus a bit less on clinical aspects, disease staging. You mentioned disease heterogeneity, but that's about it. So, are you aware of the work of Peter Jalovice, who is also a professor at our university, who also does a lot of proteomic work on Alzheimer's? And how do your data fit with what they have recently found Could you maybe comment on that?
Yeah. So first of all, it was indeed really a publication not only about finding robust biomarkers for Alzheimer's, but we were also just interested in developing methods to do these kind of meta-analysis using raw mass spec data. Right? This was not done before, so we were also again creating our own methods in that sense. Regarding the Peter Jelle-Visser data, I'm not completely up to date on his results. Yeah.
Well, he and colleagues, they actually identified different subtypes based on proteomics data. And one of the subtypes actually overlaps quite nicely with the glycolysis findings that you have. So the question there could be, could you actually be looking at one of the subtypes? That might also relate to the other question on whether amyloid and tau could also still play a role, because they might actually represent a different subtype. So to which extent do you think you are capturing a disease heterogeneity, or is it still too early based on power, sample size, etc.? So is that something that would be the next step, or do you think it's still too early to go in that direction?
Yeah, I think, I guess the group of Visser, they of course had all the clinical data related to those samples, and unfortunately one of the big missing points of this chapter or this publication was the fact that we just did not possess this clinical data. We almost had to really email the original authors, ask for it, and this was found to be very problematic. So we do not really possess the data to make such distinctions. But if you ask me if this could potentially be such a distinctive type of Alzheimer's, I would say no, because we have here data sets from 10 different data sets that show roughly this upregulation of glycolytic enzymes. And I would be surprised if they all found specifically patients that would be of such a subtype, right?
Could be. Of course, it could also be that due to the lack of power, you only find the strongest hits, which then could represent one and the same subtypes. So it doesn't necessarily exclude each other, but I understand your point of view. Then maybe I can ask your other paradigm to read out statement number 7 as my final question.
Number 7 is the field of proteomics should and will become more standardized, while data sets will become larger and more complex. As a result, proteomics researchers will become experts in bioinformatics tools.
Yeah, thank you very much. Well, as a DN or FM student, you know how to bridge fields and how to actually end up combining the best of both. But do you really think that people then should become experts, so to say, at all these different domains? So let's say in this case the biology, the technology behind this, as well as the biostatistics or the bioinformatics. Because you could also argue that if people become more and more expert in different disciplines and then talk to each other, you might end up more. So do you think this statement always holds up in that respect? Is that your experience as the end graduate, that eventually merging things by yourself as one sole researcher is the key?
So I think this is more my opinion about the fields of proteomics, I guess. Right? As we come from a time where people were still testing all kind of different methods and so forth, indeed, right, a proteomic researcher needed to be an expert in the mass spec and the methods. But just like genomics and transcriptomics, I believe at least that these methods, especially for something like clinical proteomics, will and should become more standardized, right? So then a researcher would not have to spend time developing these methods. On the other hand, right, these data sets will become more large, and then it's also incredibly important, if you ask me, that a researcher is able to handle such amounts of data, right? Previously people could do it maybe in an Excel file. If you have 5,000 of peptide information data, you have millions upon millions of rows of data. Yeah, my computer won't handle it, let's say. And therefore, I think, they don't have to become the an expert IT person, right? I'm not stating that, but I do think that some knowledge about how to, interrogate data in a tool like R, right, is of crucial importance, especially for an omics kind of type of science, where you just have so many data points that you can work with. Just looking at the statistical analysis between proteins is interesting, but maybe we want to look at the peptides or the post-translational modifications or the proteolases. And this kind of statistical analysis, in my experience, is just not very feasible with the classic click and drop kind of
I understand.
methods. So
Yeah. Okay.
that's where I'm coming from.
Thank you very much. Thank you for your answer. Also like the fact that you hinted at multi-omics being important. Of course, requires even more computational power. Don't forget epigenetics when you do that. I give the word back to the prorector.
Thank you very much. The opposition will be continued by Dr. Kuipers, Associate Professor in Mass Spectrometry Imaging in Perioperative Diagnostics at Maastricht University.
Thank you. Dear candidate, also from my side, congratulations with your really nice thesis. It was a pleasure to read. Some interesting workflows and also results in it. So, of course, I also have a few questions. And I would like to start with chapter 4, coming back to the glycolysis markers you found. There are also several other diseases, or even brain diseases, where glycolysis might be important. What would be, whether to check if this is really specific for Alzheimer's disease, what would be your next step, your data mining wise step, to find out if this glycolysis marker is indeed specific for Alzheimer's disease?
Yeah, thank you, esteemed opponent. Indeed, right, we find glycolysis in this data set of only Alzheimer patients and control samples, and indeed, right, it would be interesting to add other dementia-causing diseases or even other neurological diseases. And yeah, if you would really want to answer that question, or let's say in a very ideal case, right, I guess the only true way to really answer that question would be to really do a incredibly large-scale study over time, right, of multiple years or even decades, where you would keep analyzing samples, let's say on a yearly basis, of how patients that suffer, let's say from MCI, and how one group moves into Alzheimer's, or one group moves into frontotemporal dementia, or and so forth and so forth. And then, of course, you can answer the question how they interact with one another, which one comes first, which one comes second, and so forth.
And would you only look then at the mass-to-charge ratios, or, well, go further than only the glycolysis pathways, maybe even go further than the proteomics and see what's further down the pathways?
I mean, I come from proteomics, right? So I would at least measure the mass-to-charge ratios, because that helps us doing mass spec-based proteomics. Of course, you could go for other methodologies, maybe for these really low-abundant proteins that are difficult to measure with mass spec. But again, maybe in the future they will become measurable. And indeed, right, if you really also want to start, I think it would be useful to start adding other type of omics to such a data set. But yeah, I don't have the experience in such other type or other omics types to know which ones or to state which ones would be the most important.
Okay, and if you, last question on the other omics, if you would incorporate this in your workflow, would it require even more heavy computer power? What would it what would be the consequence of put besides the proteomics, also the lipidomics, the genomics, and so on?
Yeah, you would get to very large and difficult data sets to work with, right? I can only imagine that the amount of computational power you would require to do a proper analysis on such data would, your classical laptop will not be able to do it, right? So packing something, a work or a job like that into, running it on a high-performance computing environment would be quite feasible, or, of course, on a cloud environment, or if you want, you can also buy a lot of powerful computers and put them in your lab, right? There's multiple solutions to such computational problems.
Okay. You also described very briefly in your thesis somewhere about intensities and cutoffs. Would that be a solution rather than look at the full data set, pick out like 5 different proteins and see how the ratio change and look at the intensities. Do you think that would be a solution to decrease the computing power?
It would indeed be a solution to decrease the computing power, but I do think you would throw away a lot of data that could be quite informative. So if you would ask me, I would not go for it, right? I think, again, with a complex disease such as Alzheimer or other types of dementia, you really want to measure all these little changes, and I think 5 proteins won't be enough, right? I believe that it will be multiple panels that detect the different pathological hallmarks that we know that cause such diseases. And then if you have, let's say, for each of these pathologies, a panel of proteins that can identify and quantify them, and then figure out which one comes first, which one comes second. If one becomes stronger, it might affect the other one, and so forth. And that you really find those little details that could maybe tell you that an MCI patient will develop Alzheimer's disease or frontotemporal dementia, and so forth.
If 5 is not enough, where do you put a limit to get to a really FDA-approved method? What would you recommend? You really want to, you're nicely on the way, but what do you think is still necessary if you really want to look at the full data set? Is still necessary to have a final FDA-approved method that is confident enough to state this is mild or heavy Alzheimer's disease?
Mm-hmm. I don't really see need for a cutoff in a way, right? Of course, right? You could, of course, select 5 proteins, say that they are effective, create antibodies for them, and then use an ELISA or something, right, to make those distinctions. But I think at one point, why would you move back to an antibody-based quantification, right? Why don't we stay with just a mass spec? And then if you can really use a mass spec in a clinical laboratory, why would you stop yourself looking at just 5 or 10 or 50 proteins? Why not do work with 50 or 100 or and so forth, right?
So then coming back to my question, what is still necessary for what you nicely found in your thesis to get it incorporated in a clinical routine as an approved method?
I personally think, by now we can do, a high number of proteins in 10s, 20 of minutes, and I think it is quite expected that this number will only decrease. And if you can, let's say, start really identifying a few thousand of proteins in, let's say, 5 minutes, I think that this is, from a clinical point of view, a very nice number to work with.
Okay, and because I hear you saying identify proteins, do you really need to identify them, all of them?
Well, of course, right? There is maybe a disease where the protein not being in plasma is the clearest biomarker. But measurement of 5,000 proteins, then maybe I would use that word instead.
Okay. Also for incorporation-wise, what is very important is the turnaround time. I was wondering, how would you decrease your turnaround time, since you need quite a lot of time to do the computing? How would you be able to decrease that?
You can very quickly say something or not, you don't have to, but the time is up.
Yeah, okay. Thank you all for being here today.
Okay.
Okay.
The time for Patrick van Zalm, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Patrick van Zalm, the degree committee here present, has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Blokland is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Patrick William van Zalm, the degree of doctor and grant you all rights attached to by custom and law. As evidence of this, I present you the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Please be seated.
Okay, I guess now Dr. van Zalm, congratulations. So, yeah, my name is Hanno Steen. I was Patrick's supervisor in Boston, and it's now upon me to provide a short laudatio. And the first question that Patrick got during the defense was, why proteomics? And he gave an answer, but I don't think it was a correct answer. I think the real answer would have been, I didn't care. Proteomics found me. And if I have to think what makes a good scientist, I think there are three aspects to it. You need curiosity, you need evolvability, and you need perseverance. And I think Patrick has really shown that he has all three. The curiosity aspect is really, he was excited, he was willing to go abroad to do his master thesis. And I think as a bachelor, when you just finish a bachelor, you're too young to think about really what you want. I think it would be scary if he knew already, I want to do proteomics and this and this and this. He knew the framework, but then was basically open-minded enough to accept whatever what came upon him. And that was basically the research environment at Boston Children's Hospital, my lab, and the research questions that we had. And it was proteomics and AD. That's how he ended up there. And then he took on this project, this so-called meta-analysis of proteomics data that until then nobody had ever done. And I think that's also important when you do research. You do a lot of things that nobody has ever done. But that also means that you can fail, and often enough you will fail. That is normal, that's part of the process. And then that's also where the perseverance then comes in. You have to persevere, you have to pull through and just get it done, move on. And Patrick, during this, while he was trying to get all the data sets together, he experienced actually the really ugly side of science, of academic science. There were scientists who really tried hard not to give him the data that he wanted. But on the other side, he also really saw the shining examples of people really bending backwards in order to help him to find the data sets, going into basements, digging through cardboard boxes in order to find hard drives with the data sets. So just as an example, so he really saw both sides. And at that moment, I was a little bit shocked actually to see some of the lack of help from certain scientists. And I could have understood if he said, listen, I do my master, I move on, I'm out of this. But no, he wanted to move on. And so he came back for his PhD, kind of, I don't know, he liked me, I liked him. He liked the work environment at Boston, he liked the city. He came, and then COVID hit. And that was another opportunity to show a lot of perseverance and curiosity. And so I thought it was hard to try to do homeschooling of a second grader, but you know, Patrick's hardship during COVID was very different, was more of the emotional type. They were alone. He was one of the internationals, they couldn't go home for Christmas in 2020 and so on. It was tough on them. And luckily, we were involved in this large-scale COVID project funded by the NIH that in the US, and even though a lot of people were hanging out at home because the labs were closed, our lab was working more than ever. And I think actually was great for the lab because it really kind of provided them with almost occupational therapy. It kept them busy. And also here's now really where Patrick's evolvability came through, and also this aspect, you take something, you discover that you really like it, and you run with it. And that was a computational aspect. He joined my lab to work in the lab at the bench with pipettes and so on, but he ended up just spending almost all his time at the computer because that was his, after Teresa, his true love, his true scientific love, that he really wanted to work with computers. He didn't want to work with pipettes and so on. And that is really the aspect also of evolvability. You have to be curious, you have to test many things, and then once you find something, you have to run with it. And that's what Patrick did. And if I have to use a attribute to describe Patrick, I would use the word remarkable, actually. Because I'm always amazed by people who excel and make a career in a field that they haven't studied, where they have zero formal training in. And Patrick is one of them. He studied neuroscience, psychology. I don't even know what you officially studied. But anyway, it wasn't computer science. There's zero formal training in computer science, but that's now where he's working in, and I think he's not going to leave that field. So in that regard, it's amazing to see people who can do that. And for me as a supervisor, it's even more amazing to see when it happens in my lab under my watch. So I'm really proud of you, Patrick. Congratulations.
Thank you very much.
Dear Dr. van Zalm, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. That's the printed sentence. Of course, I'm also proud to have interacted with you in 2017 and added you to the selection of drug development and neurohealth. So I'm very happy to see this trajectory that you went through. So congratulations again to you and your supervisory team, your family. And I'm now to close this meeting, not before to have announced that we will move to the museum, it's called now, where to make a picture, yeah, with the stairs. So, okay. And then, of course, I close this meeting.